This video is a recording of the first webinar of the SGD webinar series titled SGD, a Catalyst for Biological Research. Please enjoy. Hello and welcome everybody. My name is Kevin McPherson. I'm an assistant biocurator for the Saccharomyces Genome Database, SGD. In our first talk today in the SGD webinar series, we'll be having a brief overview on how SGD can quickly and efficiently inform you about your favorite genes. Anytime throughout the webinar, if you have any questions, please submit them through the Q&A button on the right-hand side of the page through the menu. We'll be stopping by periodically to answer your questions, and we'll also be having a Q&A session at the end, so please be sure to stick around for that. All right, so what is SGD? SGD is a free community resource about the budding yeast S. cerevisiae. It provides high-quality, manually curated information on the yeast genome, genes, and gene products. Importantly, SGD brings multiple types of data, such as phenotypes, regulation, expression, etc., from different sources all into one place, which are easily accessible through locus summaries, SGD tools and resources, and the many links and annotations made across the website. By putting all these data into one place and making it intuitively retrievable, SGD accelerates research by connecting you to critical information that would otherwise take valuable time to sort through and acquire. To explain, let me draw an analogy. Let's say that you're a paleontologist who wants to study the skeleton of an extinct mammal. First, you'd have to enter an ancient tar pit and spend time retrieving the bones of this mammal. Once that's done, well, you'd have a mixed pile of bones, not quite helpful yet. You'd have to spend more time sorting the bones into meaningful categories, ribs, vertebrae, etc., in order to see how the parts make the whole. And with even more time, you can reconstruct the animal's skeleton and gain impressions you never could have from just the pile of bones. Or you could simply visit a museum, skip the tar pit, skip the sorting, and have the knowledge you're looking for all in one exhibit. So how does all this relate to yeast research? To understand a gene and its product, you'd first have to sort through the tar pit of literature, spend time acquiring relevant papers, and of course, sort through the data. The only problem for both cases, of course, is time. But like the museum, that's where SGD comes in. SGD finds papers relevant to a gene, organizes their data into meaningful categories such as gene expression, regulation, function, etc., and puts them all together into one place, like reconstructing a skeleton, to drive a big picture understanding of a gene and its role in the cell. Essentially, using SGD is like walking into a museum and finding an exhibit for your favorite gene. It's interesting, it's incredibly informational, and it's much, much better than the tar pit. To show you what I mean, let's say that while studying ribosomes, I came across the gene name RPS19A, which I know nothing about. By pulling up SGD's locus summary page on the website, I gain an immediate overview of RPS19A's description, sequence and chromosomal features, protein info, mutant phenotypes, genetic and physical interactions, gene ontology terms, and more. All of these different types of data, which are all important angles to consider this gene from, are assembled into one place from hundreds of different journal articles. Additionally, these data are annotated and interconnected with other pages and locus summaries. You can hop from place to place, gaining a stronger idea of how RPS19A is related to the overall biology of the cell. In essence, if SGD is a museum, then it's a museum on the entire yeast genome. RPS19A is just one exhibit in a whole savanna room of RPS genes. For now, let's just focus on RPS19A. For each of these data categories, here are some of the questions you might consider in relation to RPS19A. In the next few minutes, we'll go through some research questions concerning each of these categories, starting with description, and see how quickly you can answer them on the SGD website. So general questions like, what is RPS19A and what is its significance can be answered on the RPS19A locus summary page through the description and summary paragraphs. So let's enter the website now, yeastgenome.org, and check that out now. So on the top right-hand corner of the page, you'll see a search box. I'll use that to search for RPS19A. And here we are, RPS19A's locus summary page. From the description paragraph over here, we can see that RPS19A is a component of the small ribosomal subunit, is critical for assembly of pre-Fortius particles, has a paralog called RPS19B, and a human homolog implicated in the disease diamond blackfan anemia. We also see that RPS19A has 5 other aliases to which it's been referred. Now, that pretty much answers what RPS19A is and why it's significant, but let's see what the summary paragraph has to say. To find the summary paragraph, just scroll down the page, and here we are. A 2-part summary about RPS19A itself and yeast ribosomes in general. You'll notice that in this summary paragraph and throughout SGD in general, useful links are embedded in many places to help connect you to the big picture. For example, we just learned that RPS19A is important for small ribosomal subunit assembly. If we wanted to learn more about this process, we could follow some of the links here and find out more about it on another page. It's also important to emphasize that these are expert and impartial summaries written by actual biocuration scientists at SGD. Not all of the locus pages will have a summary paragraph like this, but for the thousands of genes with them, roughly 40% of the genome, they're a great resource. All right, so let's move on to our next set of questions, back to the slides, about genome and sequence data. Important genome-related questions you might ask about RPS19A include, what is its DNA sequence? What chromosome is it located on, and what are its nearby chromosomal features? Does it have any introns? So returning to the RPS19A summary page, to find the page that summarized the sequence and DNA data for RPS19A, scroll to the top of the page and find the gray toolbar with all the tabs. Here I'll click on the Sequence tab. And here we are, RPS19A's sequence summary page. Scrolling down a little bit, over here in the gene location box, we see that RPS19A is located on chromosome 15, and it also shows us the base pair coordinates of this. Below it, in the subfeatures box, over here, we can see that RPS19A indeed does have an intron. If we scroll down the page a little more, we can see RPS19A's DNA sequence. The red region here corresponds to the sequence of the intron. Additionally, you can open up this menu here to see the flanking sequences, the coding DNA itself, or the DNA without the intron, and the amino acid sequence. Scrolling down a little further, the alternative reference strains section enables you to see how the RPS19A DNA sequence varies among different strains of S. cerevisiae. So we can open up this menu and select another strain. If we scroll up to the top of the page again and follow the Genome Browser link here on the right side of the page, SGD will take us to our Genome Browser tool. So let me just open that up for RPS19A. Using the SGD's Genome Browser, you can explore the gene and any other nearby features along with other experimental data visualized in the Genome Browser. I pre-selected some data tracks that I thought were interesting, like if I scroll over to the right over here, transcription factor binding sites, polyadenylation sites that correspond with the borders of the intron, and nucleosome enrichment. This tool is a really great way to explore the S. cerevisiae genome, and there's really a whole lot more to see about this tool, but we'll go over more on SGD's Genome Browser in an upcoming SGD webinar. So that pretty much covers sequence data. Let's return to the slides and find out more about RPS19A's protein product. So here are some protein questions you might consider. How many molecules of RPS19A are in the cell at a given time? What are its protein domains, and are these domains shared with any other proteins? What are the physical and chemical properties of the RPS19A protein? Once again, if we scroll to the top of the page and locate the gray toolbar, simply click on the Protein tab to be taken to the Protein Summary page for RPS19A. In the Experimental Data box over here, we can see the protein abundance, or how many molecules of RPS19A there are per cell, and of course the source. Scrolling down the page a little more, in the Domains and Classification box over here, we can see RPS19A's domains, the coordinates of these domains, and the accession IDs to external databases. Below this box is the domains location box, which maps RPS19A's domains relative to its overall sequence. Scrolling down the page a little more to the shared domains diagram, we can see that RPS19A over here shares 4 domains, its 4 domain domains with its paralog, RPS19B, which makes sense. Going down the page even further, we can find information on the physical and chemical properties of RPS19A's polypeptide, such as its molecular weight, isoelectric point, and amino acid composition, percentage, and frequency. That covers RPS19A's protein information. Are there any questions? Well, actually, I'd like to bring up one protein data question we've received in the past is where we can find information on post-translational modifications in SGD. So let me just go over that really quick right now. I'll open up a new tab, enter the SGD homepage, and search up something like your favorite gene. So I'll type in PAH1. So let's go check out the locus summary page for PAH1. Enter the protein summary page on the gray toolbar, and scroll down to find the post-translational modifications box. And here we are. This is pretty much where you'll find post-translational modifications in SGD. If you scroll up a little bit more and look at the amino acid sequence, you'll note that SGD highlights the positions of these post-translational modifications in the sequence box. So before we move on, are there any other questions? I'll just return to RPS19A's page in the meanwhile, so I'll close that tab. All right, so let's move on to questions about phenotype. Phenotype-related questions you might ask is RPS19A essential? Do the cells die without it? And if it is not essential, how does the growth rate of the null mutant compare to the wild type? So let's return back to the RPS19A summary page, scroll up to the top, and locate the Phenotype tab in the gray toolbar. This page summarizes phenotype data from mutant experiments on RPS19A. To find out if RPS19A is essential, scroll down to the annotations table, find the filter box in the upper right-hand corner of the table, and search for inviable. Nothing. So let's try viable. So it appears that RPS19A null mutants are viable, even though the RPS19 protein itself is essential. This is likely because its paralog, RPS19B, also encodes for the protein, RPS19, but we'll come back to that fact later. What about its growth phenotype? If we search for growth, we see that RPS19A null mutants have reduced competitive fitness, reduced fermentative growth, and reduced vegetative growth. This makes sense, as RPS19A is part of the ribosome. Now, this answers our phenotype questions, but just to show you really quick, I'm going to scroll down a little bit. There's a gorgeous diagram down here that shows you which genes share the same phenotypes as RPS19A. You can filter genes by the number of phenotypes shared by using this slider down here. And of course, click on anything in this diagram to be taken to a page that shows more about it. So if you click on one of the genes, for example, it'll take you to its locus summary page. All right, so let's return to the slides and find out more about RPS19A's interaction data. Questions you might ask about this topic include, does RPS19A have any synthetic lethal interactions? Or in other words, are there any non-essential genes that you can delete in combination with RPS19A that will cause the cell to die? Additionally, what genetic interactions does RPS19A share with its paralog, RPS19B? Let's return to the RPS19A summary page, scroll up to the top, and find the Interactions tab. So if we scroll down to the Annotations box, just like in the Phenotypes tab, we can enter the search box or the filter box on the right-hand side and search for synthetic lethality. So it looks like there are 4 genes that are synthetic lethal with RPS19A, including its paralog, RPS19B. Thus, deleting both RPS19A and RPS19B depletes the cell of the essential protein RPS19, which results in cell inviability. To see which genetic interactions are shared with RPS19B, scroll down and locate the interaction network diagram. This diagram shows you the genes that interact with RPS19A and which genes interact with them in turn. So we can filter by the number of experiments used to elucidate these relationships on the bottom slider. Check that out, that's quite a lot of genes. We can also filter by physical interactions only and genetic interactions only. So filtering for genetic interactions only on this diagram shows us that RPS19A shares 2 genetic interactions with its paralog, RPS19B. That covers our interaction questions. Before we move on to gene ontology, are there any questions? All right, so let's return to the slides and move on to questions about gene ontology. Gene ontology is basically a controlled vocabulary for the molecular function, biological process, and cellular component of a gene or gene product. To give some examples, kinase activity would be a molecular function, glucose transport would be a biological process, and nucleus would be a cellular component. SGD annotates these categories for every gene. So using the Gene Ontology tab, you can find out what biological process, molecular function, and cellular component your gene of interest is involved in. So let me just return to the summary page for RPS19A, scroll up, and click on the Gene Ontology tab in the gray toolbar. So scrolling down a little bit, we see that RPS19A is annotated to a number of biological processes, including ribosomal small subunit biogenesis, which coincides with what we read in the description and summary paragraphs. Its molecular function is that it is a structural constituent of a ribosome. And scrolling down a little more to see cellular component, we see that its cellular component is that it is part of the small subunit of the cytosolic ribosome, or cytosolic small ribosomal subunit. That pretty much answers our gene ontology questions. At this point, I'd like to shift our discussion from using SGD to find info about your favorite gene to using SGD to find info on your favorite groups of genes. SGD's gene ontology annotations are one way you can do this. To show you what I mean, if we click on cytosolic small ribosomal subunit, RPS19A's cellular component, we will be taken to a page that lists all the genes annotated to the small subunit. So if we scroll down, we see that there's a table here that lists all the genes that are annotated to cytosolic small ribosomal subunit. In the middle of the page, so scrolling up, we also see a diagram that shows us the go terms, cytosolic small ribosomal subunit, and its parent term, cytosolic ribosome. If we click on cytosolic ribosome, we will be taken again to a page that lists out similar information, all the genes that are annotated to the cytosolic ribosome, as well as a diagram that shows us the go term cytosolic ribosome and its child terms, the small and large subunits. In essence, SGD can assist you in investigating groups of genes in addition to individual genes. For example, if we look at this diagram here and add all the annotations together, we see that there are roughly 180 genes annotated to the entire cytosolic ribosome. So returning to the slides, let me pose a question real quick that involves hundreds of genes. For example, how does the number of subunits compare between the cytosolic and mitochondrial ribosomes? Well, we just found out that there are 180 genes annotated to the cytosolic ribosome. Let's search for mitochondrial ribosome on the website and see how this number compares. So I'll return to the website, scroll up to the top of the page, and enter mitochondrial ribosome in the search box. Since this is a cellular component, I'll filter the search with the Go Cellular Component category, and there it is. I'll click on mitochondrial ribosome. Scrolling to the diagram again at the center of the page, we see the Go term mitochondrial ribosome and its child terms, the large and small subunits of the mitochondrial ribosome. Adding all the annotations together, we see that the entire mitochondrial ribosome complex adds up to roughly 90 annotated genes. This is half the number of those annotated to the cytosolic ribosome. Although there are many biological reasons this could be, a natural follow-up question to this discovery would be, are there any undiscovered subunits of the mitochondrial ribosome? Answering this question takes just 5 minutes by using one of SGD's tools called YeastMine. YeastMine is a multifaceted search and retrieval tool that can analyze entire lists of genes, thousands at a time. We won't be going over YeastMine in depth today, as we'll have much more on YeastMine in our next SGD webinar on May 4th, but I want to give you a brief overview of what it can do. Let's say that you have a list of genes that all have a particular function, like kinase activity. If you take another list of genes, these all having a particular phenotype, like chemical resistance, you can intersect them in YeastMine to produce a final list of genes that has the qualities of both. A list intersection would take the genes that are common to both of these lists and store them in a new list. In other words, this list will contain genes that are both function and phenotype specific. Let me show you what I mean. So I have a tab here in YeastMine. So here's one of YeastMine's page, and I've pre-prepared 3 gene lists. I have one big list of all 1,059 genes that have genetic or physical interactions with the mitochondrial ribosome. A second list of 672 uncharacterized open reading frames, and a third list of 1,146 genes implicated in respiratory growth phenotypes, since the mitochondrial ribosome, of course, would be important for growth in the presence of oxygen. It's important to emphasize that despite their size, each of these lists took just about a minute to make. I'll just go ahead and check mark all 3 of these lists. And using YeastMine, I will intersect them. So just press the intersect button. Voila! A new list of genes was created. So let me just open that up. The genes in this list as the intersection of those 3 lists of genes, therefore A, interact with the mitochondrial ribosome, B, are uncharacterized, and C, are implicated in respiratory growth. These genes have the characteristics of all the 3 lists of genes that I've intersected, and are potentially undiscovered subunits of the mitochondrial ribosome. All right, so that concludes our webinar. If you have any questions, please do say for our Q&A session. Otherwise, thank you for coming. And please make sure to keep an eye out for our follow-up email for more information on our next webinar, the SGD website, and our YouTube channel, where today's webinar will be uploaded. Thank you again for coming.
Thank you for watching the SGD webinar series. If you have any questions or comments, please contact us at sgd-helpdesk@lists.stanford.edu.